Yeah, first of all, thanks for the chance to be here and the nice invitation to this webinar and symposium. And I hope, as you mentioned already, that it's a start of future collaborations. So now, as my talk is the last one just before lunch, I try to keep it relatively light. We heard lots of exciting science in the morning already. And I promised you, so here on the cover slide, this picture of zebrafish that you see on top is the only one that you will see throughout my whole talk. So I was working in zebrafish before, but as Morgan mentioned, now my focus shifted more to working with flies. But I'm here today to talk to you more in my role as a co-director of the charity Trend in Africa. And I want to give you more of a broader overview about genome editing and efforts that are going on in Africa to promote genome editing research. As you can see, I'm not African, so I try to give you my point of view, my subjective perspective on the topic. That's based on the kind of interactions that I had with African colleagues and scientists. And also over the last years, we have a lot of discussion with people in the community doing the science in Africa, how we could actually help them to promote research in this particular topic. So just a few words about Trend in Africa. So Trend in Africa, sorry, my slide is not changing. Okay, was founded in 2011, and it's a charity whose mission is really scientific capacity building and knowledge transfer. So the idea is that we get experts around the world that know about a certain topic, and then they try to go to different African countries and share this knowledge to empower local scientists and using it for the individual research schemes. So you can find more about it at the homepage that I show you down here. Just a few numbers, what we have been doing over the last years. So since its foundation in 2011, there were more than 100 courses that happened. We have more than 900 alumni that went through these courses. We engaged with more than 10,000 members of the public, and we have partnership agreements, or we did shared courses with more than 50 universities in Africa over the last years. So there are different programs that have been running. So it started initially mainly focusing on neuroscience and using different neuroscience models, but over the years it developed in quite different directions. At the moment, also just because we heard this nice presentation about artificial intelligence, I wanted to draw your attention to this particular program here that's called Trend Carmina. That's at the moment a program that we have been running in different countries, and it's very successful. And it's about computational neuroscience and how you can use machine learning approaches in your daily work. So there was a workshop now happening in July this year, but there are more versions planned in the next couple of months and years. So just please take a look. And if you're interested in the topic of machine learning, artificial intelligence, and how you can use like hardware and software to improve your wet lab science, so I really recommend this course. Another action that the Trend has been doing, it's involved in policy making. So we try also to get in touch with local science founders and promote scientific infrastructure building. And one example I wanted to mention here, also because the plan of this group is to set up a zebrafish facility or core facility that can be used as an infrastructure for researchers around. So this is one example that was very successful and driven by one of our co-directors, Mahmoud. So he set up this biomedical science research and training center. This is a center that was established in collaboration with the local government in the north of Nigeria. And the idea was really, if we kind of provide resources at a focused location, it can serve as a hub to promote research for all people around. So what we did there is we donated equipment. It was mainly microscopes. So this is really cutting edge microscopes that have been set up in this facility. So they build a new building just for this facility. And the advantage, of course, is that it reduces the costs for organizing activities. So if you want to teach microscopy to other researchers, they can come to this particular location. It reduces logistical barriers. So it's easier to run courses once you have the infrastructure established. And of course, it's also good for the local scientific community because they know they can rely on this resource, and you generate local expertise. So I think it is very much in line what we want to propose here for the zebrafish facility, to have a similar spirit and just get people involved in setting up this local facility. So here, just a summary of what Trend has been doing. It's a little bit outdated, so this is from 2020, but you see that there's different countries where we have been active. There's alumni and courses that have been running there. There's a certain focus on Nigeria and Kenya. So this is two countries where kind of traditionally you get lots of applicants for. Unfortunately, in this map, so to me, there's still a empty spot. So looking forward to changing this in the next couple of months. And as you can see down here in the bottom of my screen, so these are the different subject areas where we have been active. So scientific writing, molecular biology, and also genome editing. So this is what I was kind of establishing in Trend, a program for genome editing, and that's what I briefly will talk about today. But before doing so, we already had these kind of very nice introductions about genome editing techniques, how you can use it in your research in zebrafish. What I just wanted to make clear when we talk about genome editing, what we're actually talking about, because I realized that often there's a little bit of confusion, and I think it's very important to be precise in the language that we're using, that we can also communicate well to regulators or the general public so that they understand what we're actually talking about. And for this, I want to make a distinction between genome engineering and genome editing. So genome engineering, the way we understand it, or the way I will use it in the talk, is the manipulation of genetic information of an organism by introduction of exogenous genetic material. So this is what we have seen in this transgenic fish examples that we saw in the previous talks, or in general, when we do transgenesis, what we usually do, we genetically modify an organism by introduction, for example, of a fluorescent marker that then allows us to track the movement of cells in the heart, or to look at the beating heart, and to visualize certain biological mechanisms and so on. However, when we use genome engineering, what is also happening is that we normally genetically modify the organism. So this would be a genetically modified organisms, and you probably know about the kind of regulatory hurdles that then come along if you have a genetically modified species. Instead, when we talk about genome editing in the kind of stricter sense of the meaning, genome editing is targeted manipulation of a gene of an organism without introducing exogenous material. So this can be via, for example, this base editing, what Vincenzo just mentioned. So this is just the exchange of a single nucleotide. And this means that we can manipulate the genome, but we don't leave any traces of this manipulation. And for both, as we already heard before, we can use the CRISPR-Cas9 system. So what I want to do now in the next couple of minutes is just to give you some examples how genome engineering and genome editing can be used in the African context. And for this, as I mentioned already before, this is the work of a consortium of scientists. So we came together just discussing about genome editing and how can we promote this technology on the African continent. So this was a group of scientists from 12 different countries. So you will see that this is kind of trying to represent different areas on the African continent. They were not only from academia, but they were also from industry, and mainly 3 different fields. So this was control, because as I show you in a little bit, so this is probably where we are most advanced in the context of genome engineering. Then in crop and livestock, because there's a huge need to develop these tools to improve phenotypic traits of crop and animals, and then also in the context of medicine. But we also had some people that are working more in the regulatory space and in international agencies, because they have a better idea of not only taking the scientific perspective, but also coming from the regulatory side, what is important if you work in genome editing. So if you're interested in the topic, I recommend you to read this article. So what we did there, we state the potential of genome editing, and then we also try to identify roadblocks, why this innovation is not really taking so much traction as it could on the African continent, and recommend actions. So just as a brief summary, what we discussed and what we show there, what are the main applications for genome editing or genome engineering in Africa? And I would say, from my point of view, so the most 2 urgent problems that have to be faced is, on the one hand, food security. So here on this map, you see just which countries are kind of suffering most from food insecurity. And I think it gets pretty clear that Africa is most severely affected by this problem of malnutrition and lack of stable food supply. And on the other hand, public health. So if we see here on this map, the global burden of diseases, this is also extremely high in Africa. So I think that these are really 2 areas where scientific research and bio-innovation and genome editing can have fundamental impact on the well-being of the population. So what I want to discuss in the context of food security, what can we do there? So there's different ways how genome editing can help. And these are approaches that have been used in different contexts as well. So for genetically modified organisms, also try to provide the same end result. So we want, on the one hand, to enhance the crop resistance. So if we have crops that grow in a certain environment, we want to make them more resistant to drought, heat, and pests. So ideally, we have a higher yield. So we improve food security and reduce the reliance on food imports. So if we can stabilize food supply in Africa, then this would be a major positive outcome for the local population. In a similar line, we can also try to improve the nutrition. So we can try to engineer crops that have an increased vitamin or mineral content. So like this, they provide kind of fundamental nutrients for the population. We can try to increase the productivity. So we can try to reach higher yielding crops that can contribute to economic growth. And the same what is true for crops, of course, applies also to livestock. So we can also try to improve the livestock and get more disease-resistant livestock, so that increase the productivity and the economic benefit for farmers. So this, of course, has not only been recognized by us. So this is, I would say, the most intense area of research in the context of genome editing. And there are some spectacular like breakthroughs that have been done in Africa. But just to show you what's the difference of genome editing, kind of a classical approach, just taking a step back, because I think many of you are probably not working in the crop field, neither do I. But from my understanding, the classical way of improving the phenotypic trait of a plant, here's just an example where you see an elite variant that is, however, producing a lot of grains, but it has a high disease susceptibility. So what we can do in a classical breeding approach, we take this elite variant, we cross it to a donor variant that is disease resistant, and then in the subsequent generations, we just select for the beneficial trait for the resistance of the disease, combined with the high production of this particular crop. However, this is a very time-consuming step because it takes up to between 8 and 10 years from the initial variant to this improved variant. Then what we can also do to kind of, if we don't have this disease variant as a potential donor, we can just use mutagenesis, and this is a kind of classical breeding. So we mutagenize our starting plant via chemicals or radiation, and then also select and backcross, what is also a very time-consuming process. There's then the kind of more established approach, what has been used in the past. And this is, again, for this, I wanted to clarify the difference between transgenesis and genome engineering and genome editing. So here we can introduce a certain trait via genetic methods, for example, some plasmids, that then alter the traits of the transgenic plants. However, this is then a genetically modified organism and needs certain regulatory steps to be used as a product in food production. And as a last step, what nowadays I think people favor, or what is the huge potential of genome editing. So we can use CRISPR and the Cas9 system to modify specific traits of the plants. However, there's no trace of this manipulation in the variant, and it will be the same product. Then it depends on the regulatory landscape if this is considered to be a genetically modified organism or not, and I will talk about this a little bit later as well. But we call this precision breeding because in the end, we have the same outcome as from classical breeding approaches, however, using novel genome editing tools to get to these results. And as you see in the timeline, so this is substantially faster. So it's from the initial step of experimental design to a product that can be commercialized, it's only 2 to 5 years. And excitingly, this has been done in an African context. So there's like African scientists that are kind of pushing forward to use local plants and improve their disease resistance. So here's an example of sorghum that has been gene edited to be resistant to Streba plants. So this is a nice example where genome editing has actually achieved an immediate outcome for this particular species. If you're more interested in this topic, I recommend you to read this recent review. And you see here the different waves of genome editing. So what has been used over the last years, or has been developed in the last 10 years from different African scientists. So what I want to mention here specifically is the associations or two consortia that really push forward the development of genome editing tools in their respective home countries and more globally in Africa. So this was the one example that I wanted to mention in the context of crop. And the second example I want to briefly touch on is in the context of public health. So there we can use genome editing to understand better how pathogens work. So we can do some basic research. We can also work in the context of vector resistance. So try to understand if we have, for example, mosquito populations, what changes happen in the genome to make them resistant to classical vector control mechanisms. And we can study disease-causing gene variants. So I think this is getting very interesting in the context of personalized medicine, a little bit what Vincenzo also mentioned before, that we can develop novel strategies that try to copy individual alleles that are present in the population and use genome editing for this. However, I think these approaches are still very far in the future. It will still take some time until they are fully developed. However, the most advanced in the context of public health is a genome engineering method that's called gene drive technology. So I don't want to go into too much details, just as a very rough summary of what gene drives is doing. So gene drives are genetically engineered agents that can break Mendelian inheritance. So if you look at Mendelian inheritance, usually if you have a mutation in a certain population and we cross them to the wild type, as you can see here in the top scheme, then we have a 50% chance that the trait is arriving in the next generation. However, gene drive inheritance is able to break this 50% Mendelian inheritance and leads to nearly 100% of the offspring that inherit a certain modified gene. So this was a method that was initially established using zinc fingers and this other endonucleases that we heard about in the morning. But now with CRISPR-Cas9, the efficiency of these tools is increasing tremendously. And so there are two different ways how these gene drives can be employed. So the first is for population suppression. So this is the idea is that we target this gene drive into a gene that's essential for the population. If this gene drive then is spreading, it leads to the collapse of the population and we can eradicate, for example, a mosquito species. However, I think in the last years, people realized that this might be a bit of a dramatic approach or drastic approach and rather talk about population replacement strategies. So what we can do here is one, design the gene drive cassette with a certain effector gene. And then this is taking over the population. So we don't completely eradicate the initial population of mosquitoes, but they have some altered traits that maybe in the context of malaria, they might not be able to be infected by Plasmodium. So if you're interested to learn more about this, there are several initiatives that work on genome engineering and gene drive technology. And I think one big breakthrough was last year when there was the first transgenic mosquito made in Africa by Transmission Zero. So this was in Burkina Faso. So there really they set up the facility to be able to generate these tools in their own institutes in Burkina Faso. So I think that was really a major step forward. There are other consortia like Target Malaria or the Pan-African Mosquito Control Association that are also involved in this space. And just next week with Trent and also with Vincenzo that's involved there, we are organizing a small symposium on CRISPR and gene drives together with CRISPR Medicine News. So you can still sign up for this webinar. So you want to learn more about gene drive technology and how this can be used for vector control, especially in the context of malaria. I encourage you to check out this webinar and join us next week. So far, what I tried to show you in these two examples is that there's really tremendous potential for genomic technology in Africa. So there's really the kind of big challenges that the continent is facing. We think, or I personally think, that genome editing and genome engineering or genomic technologies in general, they are an important step to solve these challenges. But there's still a long way to go. And this is also something that we learned when we were analyzing the landscape. So we looked at the number of patents. So as a readout of bio-innovation. So how many companies are developing intellectual properties around CRISPR and gene editing technologies. And you see that since the establishment of CRISPR, the kind of number is growing exponentially. However, when we then look where these patents, where were they filed, so in which countries, what you can see here in 2013, mainly in the States and in China. And then over the years, other countries took this innovation, also took this up and also started to develop. However, unfortunately, if you look in 2023 and the map of the world, in Africa, there was still a little adaptation of this technology and little patents that were really coming from an African context. So here just the numbers for the whole African continent, and you see that they are still relatively low in compared to the rest of the world. So this is not only reflected in the number of CRISPR patents, but also as scientists, you know, for us, the currency is always publications. So we also looked at the number of publications, and also there, unfortunately, we could see that the number of African authors on publications about CRISPR is still relatively low. And what we also analyzed, we wanted just to see at which position are they in the author list. So are they in the last author positions where usually the research is conducted, or in any other position? And you see that especially the last authors in Africa, they're still like underrepresented. So I think there's still lots of work to do to enhance the contribution from Africa itself. So what are the roadblocks? So then we try to understand and discuss what keeps these bio-innovations really taking off in an African context. And the first major roadblock that I want to mention here is the regulatory framework. So with regulatory frameworks, I mean two different aspects of that are important for innovations. First of all, biosafety. So I told you at the beginning, so there's this difference between genome editing and genome engineering, but in the regulation of each country, there's no clear guidelines that distinguish between GMOs and genome edited or precision breeding organisms. And this is not only the case in Africa, so this is actually also in Europe. There's ongoing discussions because the regulatory frameworks, they just have to adapt to this new technological possibilities and make clear how do they regulate genome edited organisms. However, what's also a problem in Africa, what we saw is that there's a bigger fragmentation on the regional level. So each country has its own individual genome editing regulations. What is a problem then if you want to establish trade between countries, it's unclear how crop that is developed in Kenya, for example, might then be regulated in another country that is trying to import this. And the second aspect of regulatory frameworks is the intellectual property. So for people that try to invest in technology, it's difficult to know how will the intellectual property be protected. So there's a certain regulatory uncertainty and lack of enforcement that blocks innovation because people are not sure if then they will carry the fruits of their investments if it's not clear what's happening to the intellectual property. And this is just a map of Africa. So this was the state end of last year. By now, there's a few more countries that updated the guidelines, but you can see that actually the countries that have the most advanced guidelines or where there is a clear regulatory framework, there's also where most research is happening. So we think that these regulatory frameworks are really important, that people know in what they can legally do and how this is regulated in a certain context that they operate. Then I want to mention two other roadblocks that we identified. So this was the lack of funding and infrastructure. So I think this is a general problem, but it's even more severe in the context of genome editing because you need usually a molecular biology lab infrastructure to run genome editing experiments. What is relatively expensive to set up and to maintain. There's supply chain issues. So often there's no local production of reagents, so you have to import, and this leads to long delays and high costs. And then there's also a lack of research investment by local governments. So I think for this, it's also important that us as scientists, we talk to regulators, we talk to governments and show them that these are the problems that have to be faced if countries really want to benefit from this genome editing revolution. And a second aspect is mistrust. So especially in the context of genetically modified organisms, there's a lot of uncertainty, there's ethical concerns. People afraid, they don't really understand the technology well. So I think it's also important to organize events like this, that we educate ourselves, and then we can take this knowledge and bring it to the lay public, that they know actually what genome editing is and how it can be used to their benefit. So I think overall, I'm a strong advocate for the training of scientists, communicators, and regulators, because these are really the kind of key drivers of technological development. So who is doing this at the moment? So I just thought I do a little summary of the initiatives that are happening in different countries that try to promote genome editing research. And I mean, we are all here because of Learn and Win and Mohamed. So it's here we have like an opportunity to promote genome editing research, or to talk about genome editing research in the context of the symposium. Then, as I mentioned, so Trend is doing workshops and also networking activities. In the last years, we did a lot with FNIH. So there's an initiative that's called Gene Convene. They also run workshops mainly for regulatory, like for regulators, because they want to help in informed decision making. Then there's the African Union Development Agency, with whom we have been collaborating in the past. They set up a community of practice of people that use genome editing. So please go to their homepage and see that a good source of other colleagues that might be operating in the space. Target Malaria, I mentioned before, they do workshops and conferences specifically in the context of genome engineering. And then the Alliance for Science, they do workshops and networking. And for this, I want to draw your attention to an initiative from the African Orphan Crop Consortium and the African Plant Breeding Academy. So they are really advanced in promoting genome editing and trying to train scientists for a longer period of time. So the aim is in the next years to reach 200 scientists that go through the training program. So now they go through the third round. So they already trained more than 25 participants in the last years. And there's a call at the moment to be part of the next cohort of scientists that will benefit from this training. So if you are operating in the plant space, so I really recommend you to try to join this community there. So then, just as a last thought, so together with these people, we have been discussing how can we promote genome editing in Africa. And I think it's a very similar spirit as which we are here today. So I wanted to present you one initiative that we have been starting, actually together with Vincenzo. And this is a new association that we just founded that should promote genetic research, not only in Africa, but what we really strongly believe in, that, we need collaboration across continents and across scientists to make this a success story for everybody involved. So what we really try to do here is to bring all these different ongoing initiatives that are happening mostly in isolation together into a global network with a strong focus on Africa, because that's where we have most experience with and most of our contacts. But the idea is really to form a network of scientists that connect these individual players and then try to promote genome editing, because we think that the more people are involved in this, the more powerful we are in bringing these ideas forward. So we hope to go online soon. So we are still in the planning phase. However, if you're interested in this and you want to be part of it, please get in touch. You can write to Mohamed, myself, or just stay in the discussion and get in touch with us to know that you want to be joining this initiative. So just to sum up what I've been telling you now in the last 20 minutes. So I really think that genome editing is a relatively simple technology. You need basic molecular biology, however, to make it happen at your location. It has the potential for high impact in bioinnovation to solve African problems. And I think the key to make this happen is really collaboration, networking, communication, and capacity building, so that there's enough critical human capital on site. So there's enough experts that they know how the technology works, and then they can make this really happen in the individual locations, in their specific settings. They know best what are the challenges they have to face and how to make this happen in their particular research environment. And as I said before, so we try to follow this mission with Trend and now with Gene for All. And if you're interested, then please get in touch. And with this, I want to thank you, and I hope to see many of you at the hands-on workshop next year, and happy to answer your questions. Thank you.
Well, thank you very much, Tom. It was very important for us to know all about all this initiative and about the main idea of trend and now hopefully Gen for all. So yeah. So any question from the floor before I go myself? Before I go for question.
One comment to
Yes, Marco, please. Yes, yes.
I congratulations by the speaking because it's very extraordinary conference. I would like to view title edition genetic in Venezuela. What is your opinion?
Well, so to be honest, I don't have much experience in South America. So I think you're the expert, so you're there. I'm not sure what is the kind of research infrastructure. I think what is important is that if you want to initiate, genome editing, you have to make sure that you navigate within the kind of regulatory guidelines that are set at place. Um, and then just find the critical amount of people that is excited about it. And the major challenge that we always face is funding. So I think this is a problem um, in every country. You need to convince somebody that they give you money to do this type of research.
Yes.
But I could see that, if um, if you have a concrete application of your research, then it might be even possible to recruit private investors. And I think that's also something that's a bit underexplored currently. And for this regulatory or intellectual property protection is very important because maybe Merck can comment on this. But I think if a good regulatory framework is in place, then also it's easier to get investment into a certain
Thank you
technology.
very much, Tom. Thank you very much, Tom.
Any other question, maybe?
I don't know, Vidit, do you have
I have a
some
question
thoughts?
for Tom.
Yes, I mean.
Thank you, Tom, for your intervention. It was really rewarding. It's actually more of a comment than a question. With regard
Mhm,
to the regulatory bottleneck, UK and
mhm,
USA has already approved CRISPR-based therapy for sickle cell disease. So I
mhm.
don't know why we couldn't push forward to implement CRISPR in husbandry and plantation too.
So I think a major challenge there, from my understanding, is also, regulators are maybe not well informed. So it's or maybe I'm saying this wrong, but I think that it's a relatively new technology and has relatively like high potential impact. And I think if regulators don't understand it, there's the tendency to rather be more strict in the regulation. So I think there the major task for scientists is really uh, to advocate for it and to educate and to help to overcome this mistrust just by informed decision making. And I think there is where, every one of us has a certain responsibility or opportunity to promote knowledge in his kind of surroundings.
Yeah, yeah, no doubt. Actually, we have just to keep in mind that it's like a really teenager technology. It's like 10 years or 12 years since we start like dealing with new. I mean, it's still in a lot of minds, like it's still science fiction, as I wrote, I think in some comments. So it's still for a lot of scientists, still a science fiction stuff, but actually we just need to learn more and just need to educate, as you said, our local and our neighbor and our locality and our continent, country, for such an powerful technology for the best of all. Just a curiosity. So you said that you found some CRISPR-Cas publication from Tunisia. Myself, I know two. So do you have an idea about the other publication? I know that from Institut Pasteur, there was a team working in CRISPR in Candida albicans.
I could go back to the list that we compiled back then and see what was there as a hit for Tunisia. If you're interested, I can take a look and send it to you in case. So the way we sorted this is, we just looked in the affiliations for the affiliated institute. So it could also be that a certain author has multiple affiliations and the research was done potentially elsewhere. But I can take a look at it.
Okay.